The most important thing to come out of the modern synthesis was population genetics, which is really take genetics and make it math. Population genetics is the study of the distribution and changes in allele frequency within a population. There's a lot of words here, but let's focus like we're looking at a population. So we're getting some ideas from Darwin, but now we're looking at how the genetics is changing over time within that population. Under this field, we have a very specific definition of evolution. Now we define evolution as the change in allele frequency between generations. Remember, this is the population genetic definition of evolution. Different fields will have slightly different definitions because we're using different types of data. Population genetics is specifically looking at allele frequency. This is a little oversimplified, but because it is simple and it is a quantifiable measure, it lets us look at evolution in very specific ways and use numbers to model how evolution happens and changes over time. With this model, we get something called the forces of evolution. A force of evolution is really anything that can cause a change in allele frequency within population. And we have four: mutation, genetic drift, gene flow, and selection. Let's look at these one at a time. First, we have mutations. A mutation is simply an error in a genetic sequence that is not corrected. If you take a molecular biology class, you'll learn that there are many mechanisms within our cells to identify and repair damage in DNA. And most of them are repaired before that cell divides. But every now and then, there's one that isn't repaired before the cell divides, and that becomes a mutation. We have a couple words that we use. You see the word de novo, that's just a fancy word for new. The special thing about mutation is it creates new variation, and it is actually the only force of evolution that creates new variation. There are many types of mutations here. What you can see here as an example is a point mutation. We've just changed one single base pair, or a frame shift mutation. In this example, we have taken out one, so we have shifted the frame for our codons, and now we're actually reading different proteins. If these words are a little are confusing, make sure and go back and review the molecular biology lecture. One specific idea within mutation is Muller's ratchet, of course, proposed by Muller, and this is the idea that there is the slow accumulation of deleterious alleles. This does happen more likely in parts of the genome that don't recombine, so this means asexual organisms, the Y chromosome, and the mitochondrial DNA. By chance, most mutations are probably going to be harmful. It is easier to mess something up than to make it better. So that's why if you look at something that doesn't recombine, you're probably going to find a lot of deleterious mutations. However, this brings us to our next point of recombination. You will see some textbooks list recombination as a force of evolution. Technically, it isn't. But what it does do is it puts together new combinations of alleles, and in some ways that is just as important because with recombination, now we're considering how genes interact with each other. This recombination may be good or bad. And it's also interesting that the rate of recombination isn't uniform. There are certain spots in our genome that we call recombination hotspots because recombination happens there a lot more often than it does in other places. So again, this isn't a force of evolution, but this is how we think of how genes interact with each other. And in some ways, it's just as important. Most people have heard of inbreeding depression before, but there's also the opposite, outbreeding depression. So this happens where there's a drop in fitness due to breaking apart favorable allele combinations when you reproduce with someone from a different population. So this is what happens when you break up things that were useful through interbreeding, generally through interacting with recombination. But let's talk about our next force of evolution, genetic drift. In the most simple way to explain it, genetic drift is simply random changes. You want to get a little bit more specific, it means stochastic sampling error. In an infinite population, we would expect to see a certain certain things happen. If you flip a coin 50 times, you expect to see 25 heads and 25 tails. But if you only do it 50 times, you're going to see a little bit of deviation from those expectations. If you flip a coin an infinite number of times, you will get half and half. But we're not doing it infinite times, and we're always going to get a little bit of deviation. And that's what stochastic sampling error means. This is just a product of the fact that we have finite populations, and that population size is going to have different effects. Genetic drift, it reduces variation, and alleles can become lost, so they're no longer present in the population, or fixed, they are the only allele present in the population. Here's an example. Thank you, Khan Academy, for this lovely graphic. So in our first population, we have dark and light beetles. Due to chance, something happens and only the light beetles survive, so the light allele is now fixed and the dark allele is lost. This is something we see under genetic drift. It tends to be stronger in small populations. This is a really fun graphic here because we're looking at a population with the same parameters except for the population size. So first we have an N of 20 or 20 individuals in our population. You can see the allele frequency changes wildly, but it's a little less wild with 200 individuals in the middle and hardly any deviation with 2,000. This is a classic population genetic graph where each line represents a different independent population. We do this in simulations just to make sure that we are seeing a true trend because you want to do the same thing multiple times when you're simulating something. Because you want to make sure that you didn't look at the one simulation that was weird. With genetic drift, there are a couple of specific examples that are so special that we've given them extra names. The first is the bottleneck effect. This is where there's a temporary dramatic reduction in population. Generally, a natural disaster killed off most of the population, but after that natural disaster is over, the population is going to recover. And we do like to visualize it through an actual bottleneck. So here we have our original population. We are showing genetic diversity with more colors. So there's a lot of different colors in our original population. By chance, only a few survive. We can see we only have yellow and blue surviving. So in our new population, it will go back up to its large population size, but we've lost most of our variation. So we have a big population with very low genetic variation. We see this today in elephant seals and cheetahs. We know what happened in cheetahs. Us. We did that. That's partially through hunting and habitat destruction. With elephant seals, we actually don't really know what happened there. Um, they're doing fine right now, their population size, but they have pretty low genetic diversity. So this indicates that there is a historical population bottleneck for one reason or another. The other really important type of genetic drift is a founder effect. A founder effect is similar to a bottleneck, but there's slightly different things going on. So here we have an original population, and just a few of those individuals, they moved somewhere else and found a new population. Because only a few people are doing that moving, there's not going to be as much genetic variation in this new population from our old population here. Again, in this graphic, it is represented through color. More colors mean there's more genetic variation. Fewer colors means there's less genetic variation. This is very common when we're trying to visualize genetic information. So if you ever see any images like that, that's what they mean. The next one we want to talk about is gene flow. This means there's admixture between different populations, or someone is moving from one population to another and having children in the new population. Gene flow can be a little bit complex, but in general, it makes populations more similar, and it prevents speciation or two populations becoming different species. There are many different models for how gene flow can happen, and it really depends on what this situation is. Which population is bigger? How many populations are interbreeding? How far apart are they? So modeling gene flow can be very complex, and it really depends on the situation what exactly is going to happen. But in general, all the populations will become more similar. The last one we want to talk about is selection. So going back to classic Darwin here, now we're talking about differential survival and reproduction. Some individuals are more successful due to the hereditary traits. I am specifically just saying selection at this point because there are several different types. There's natural selection we talked about, there's artificial selection where we have selected on domesticated plants and animals, and then there is sexual selection. Selection, we are actively removing deleterious phenotypes from our populations. There is occasionally diversifying selection, but that is very rare. Again, this is not survival of the fittest. Think of this as reproduction of the good enough. We are simply removing the worst phenotypes from the population rather than just simply selecting the best and creating the strongest phenotype. That's not really how evolution works. Remember, there are three other forces of evolution that are also operating, and that is one of the reasons why evolution is sometimes so random and unexpected. So, can you explain? What does a force of evolution do? And what are the forces of evolution?